Okay, today is Wednesday, April 22nd. I was just looking on the lab schedule and noticed we have our next lab quiz next week, and then our final lab quiz the following week. So I'm thinking probably post those on Monday, kind of like we did this past Monday, right, for the last lab quiz. And I have here in my notes, we had it open from noon to 6. Do you guys recall whether that was the times? Yeah. I think I've written a million things down here, so I want to make sure. So what we'll do then is plan to have next week's lab quiz and then the following week's lab quiz on Monday. And that would be the 27th and the 6th, respectively. And that'll be open in Blackboard from noon to 6, like we did this past Monday's lab quiz. Okay. So I'm going to make a note here to send out reminder. Okay, any questions on that?
Next one's going to be on what exercises?
The quiz next week will be over 45 and 46.
Okay.
And you should look over 37 and 38, which is what we're doing next week. And then the next following week's quiz on the 4th will be over 37 and 38. And you should check out the 3 that are scheduled for that week.
Okay.
Okay. Any questions on that? How about questions on the lab exercises we were scheduled to do this week, which was respiratory organs, breathing, and respiratory volumes, 45 and 46? Anybody have any questions about those exercises or anything that I posted on Blackboard related to 45 and 46?
This is probably a stupid question, but what exactly does the uvula do again?
The uvula is really there to help direct food in a inferior sort of direction downward to the pharynx.
Okay.
Yeah, that would be really its only function. It has nothing to do with sound production. Of course, that's the vocal cords. And I'm not even sure if the textbook specifically lists that as a function of the uvula, but that's in essence what it does. When you swallow, food hits that little punching bag, right? That's what that is. It can only go down. If you didn't have it, you could get more upward movement of fluids and food up into your nasal cavity. You know, we've all experienced a time when we're drinking a soft drink and somebody tells a joke and it comes out our nose, you know. I was watching a FaceTime or Facebook video that I'm sure you've all seen of the girl that puts the Mentos in the bottle of Coke. Have you seen that?
No.
Oh my God, it's hilarious. About 2 seconds after the Mentos, you know what that is?
Yeah.
Hits the Coke. She puts her mouth over it because she knows what's going to happen. And the bottle fizzes and her cheeks get big and out sprays the Coke from her nostrils. And then she can't contain the pressure and she opens up and everything goes flying. It's really funny.
That must sting in your nose.
Oh yeah, absolutely. I mean, she didn't look like she was very happy about how it turned out. Yeah, it's a pretty funny Facebook video. Just, you know, or Google it. You know, it's been around for a while apparently. I just noticed it the other day. My wife said, "Oh, I've seen that several times." So back to 45, 46. Any questions on those lab exercises.
I had a quick question. I was looking over some diagrams and it was showing the vocal cords diagram.
Uh-huh.
There's something called true vocal cords and false vocal cords.
Okay.
What's the distinction?
Okay. So let's pull that PowerPoint up. So I have got to, let's see how I do this.
Did you happen to see Melissa's comment in the chat?
I thought it was 12 to 5, but that it might be. I feel like that's really not good though. I don't understand what you're saying. I know we had scheduled it from 12 to 5 initially, and then I have here somewhere 12 to 6. I don't know, an hour is probably not a huge difference, right? You can log in on any time between 12 and 6. So Melissa, I'm not sure I understand what you're writing. 12 to 6 is fine, isn't it? Melissa, hello.
Oh,
Okay, gotcha. All right, hang on. Let me, I gotta navigate through this system here a little bit. I got the microbiology stuff pulled up. I want to pull up the A&P stuff. And we want to go to respiratory system. Let's see, which one is it? Oh, let's just do this. Let's just do this. Okay, so let me try to now share that PowerPoint. You all see it?
Yep.
Okay, so in terms of differentiating the true from the false vocal cords, this is one of several diagrams that illustrates the more superior structure here called the false cords or vestibular folds. So they would lie just above or superior to the true cords. And between those true cords, remember, is the space we call the glottis. So that's the primary difference.
Okay.
A superior pair of structures called false cords or vestibular folds, and the true cords containing that elastic tissue that has to vibrate as you make sounds lies just below that.
Okay. I think I was filling out the lab book, and it was a top view, so I couldn't really make it out what it was.
Okay, yeah, I agree.
That view makes much more sense.
Hard to draw that, you're right. So in this lateral or sagittal view, we're looking here at one of the two false cords. This would be on the left side.
Okay.
And one of the true cords down below it. It's really, really difficult to draw. And they really should have this colored white, not red. Red sort of infers that there's skin and blood vessels there to make it that red color. True vocal cords themselves are just stretchy connective tissue. But the position is obviously correct here. I'm not sure that I mean, I see why they did it, but it's hard to tell the difference between the two other than the relative location, one above the other, right?
Yeah.
You're thinking about this view, maybe,
Oh yeah.
where we're looking down from above, right?
Mhm.
And so the true chords, as I mentioned earlier, are shown here as white, and they are a little lighter in color. And then here, of course, in the actual photograph, are the true chords here, leading down into the trachea, and you can see the C-shaped rings of cartilage there, right? See that?
Mhm.
The false chords, they have a label here, but what does that really show? I don't think it shows much of anything. So I agree with you. I think that's very confusing.
Hmm.
I would just know that they lie superior to the true chords. They're just above them. And they play no role at all in sound production, the false chords. That's the name false, right?
Okay.
Yeah. And you might remember me mentioning in the past how that first video that I showed you guys in lab, the very first day in lab, called The Incredible Machine, the Incredible Machine. It had the woman singing the scale. She was standing next to her voice teacher. The voice teacher was playing the piano, and she was looking away, singing the scale. Remember that? Ah, 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 ah. And then it showed the camera looking down just like this, and you can see her chords vibrating and the distance between the two kind of varying depending upon what note she was hitting. Do you remember that little clip?
Yeah.
Yeah. So that's what you're looking at basically in the lower photograph.
How can you sing when that thing's camera's in your throat?
Gosh, yeah, I know. That's got to be really hard. Yeah, because how would you not gag,
Yeah.
Of course,
You have to have gag reflex.
with today's technology, the camera could be really, really, really tiny. It wouldn't be quite as bad. But when that movie was made back in 1973, the camera was probably, really large. So I don't know how they did it. She had a big mouth. I don't know. The one thing that I just thought of as we were kind of fielding questions about the lab, did you guys all see in Blackboard, I had posted, and I'll try to pull this up, a handout that was, it was called the Respiratory Physiology Experiment. Did y'all see that?
Mhm.
Yeah.
Oh.
I'll pull it up. Well, actually, it's already in the folder for that week.
Did you say respiratory physiology experiment?
Yes.
Okay.
I'm not sure exactly what you guys are seeing.
Is this the one that you had us run up and down the stairs last
Yes.
semester?
Yes, that is right.
Okay.
That is correct. Give me a second here. I don't know why it's doing this, but it is. I'm just freeing up these files here. But did you guys get in there and you know what I'm talking about? Sarah's already done this, so that's why she knows.
I don't think I saw it on there.
Okay.
It was under the stuff to print off or copy and bring to lab each week.
Right. All right, what the heck's the matter here? You guys still there?
Yep.
Okay. All right, so where is it? Oh, I'm so confused when it comes to trying to share files. What I think should be there is not there. Why is not there? Okay, do you see where it's showing here the exercise 45 and 46?
Yes, and I see it now on my own computer.
Okay, so this is in the file stuff to copy and bring to lab each week, which is from the content folder. Do you see the content folder now or no? Yes?
Yes.
Okay,
Yeah.
so you've all been here, right? So here's the file, stuff to copy and bring to lab.
Yep.
And then you come down here, and then here we've got respiratory organs. Now, this says torso model. We're not using the torso model, obviously remotely like this, but had we met face to face, and this is why this was hidden from you, there's no point in me giving you this because we don't have the model to look at. So that's not something you need to worry about. There's a accompanying key here for that. The larynx model, same thing. Although I'm trying to remember, did I post in Blackboard? I think I might have. I'm trying to remember. I gotta go back and look now. Well, a little YouTube video on the larynx model. Do you guys remember seeing that? Have you looked for it? No?
Which video?
No, I haven't
I
looked.
almost,
In the lab for this week, I just can't remember if I posted any videos. Okay, here we go. Here's respiratory organs, and yeah, this is what I'm looking for. Yeah, so there's the model right there. Okay, and I posted here the larynx model that we would have used had we been in class, and it's the same model that the video shows. So you need to understand where these structures are relative to this model you see. Do you guys see the video that I'm pointing to?
Mm-hmm.
Yeah.
Okay, okay. So you need to print this off. You need to look for those structures as pointed out on the video. And there you'll see the true and false chords that Joe was asking about. This video will use a similar torso model to the one we used in lab, and we'll cover the upper and lower respiratory tracts. So this likewise would be worth watching and then relating to the diagrams that I pointed out here in exercise 45. This video is a voiceover I did some time ago relating to respiratory volumes that we talked a bit about on Monday. This is another video that talks a little bit about that use of the spirometer to measure various respiratory volumes and capacities. Okay, are we following me?
Mm-hmm.
Okay. Now here's the video that or the experiment that I was referring to a few moments ago that I thought maybe, I should draw your attention to. This is what we would have done had we met face to face. Have you looked at this at all yet? Other than Sarah?
No.
Okay. This is on the quiz next week, just FYI. So you need to carefully read through this. And I mean, this isn't Calc 3. This is not that difficult, but you need to read through this and understand what's being presented here in terms of this chemical reaction, which is also described in the textbook, actually. All right, and imagine yourself going through this simple experiment. And I give you some data down here at the bottom, which is not unrealistic. had you done it yourself. Okay. Now this video, the bromothymol blue, is a similar chemical reaction to what you would have seen had you done that experiment that I just referred to a few moments ago in that previous document. When you exhale through the straw, what are you bubbling into the fluid?
Carbon dioxide.
Right, CO2. And that CO2, as the handout talked about, when that combines with water, what you end up producing is something called carbonic acid. Do you see the handout in front of you? Where it says respiratory physiology experiment?
Mhm. Yeah.
Okay, so here's the CO2 that you're exhaling through the straw, combines with water in the beaker to form this compound called carbonic acid. That's what's shown there in gray, carbonic acid. The carbonic acid molecule in turn breaks into hydrogen ions, and these are called bicarbonate ions. Now we're most interested in the hydrogen ions, which is why I underline it, and I talk about it here. What do you know happens to the pH of a solution if the hydrogen ion levels go up?
pH would probably go down.
Right, pH drops, exactly right. And that pH drop is something that you would see in this bromothymol blue, even though it looks green. You would see a color change take place as a result of the pH falling. Okay. In the case of this experiment, we would have used a different pH indicator than this. We would have used something called phenolphthalein, and that would have turned the beaker pink, a really pretty pink red color. And as you exhale through the straw, that pink goes colorless as the pH drops. So it's just a fun little experiment. I used to do it when I taught physiology labs in grad school, and I adopted it. I kind of stole the idea. And what we would have done had you been in class is you would have worked with a partner. You would have set up these various solutions in a beaker, water, some sodium hydroxide, which is a base, and a couple drops of phenolphthalein, the pH indicator, and you would have started off with a pink solution, and you would have had your neighbor exhale through the straw while you timed how long it took to go from pink to clear. And then you would have switched roles, and he or she would have exhaled to the straw, or you would have exhaled to the straw while they kept track of how long it took for you to do it. And then you would go out and run up and down the set of staircases 2 or 3 times and come back into the lab, and your partner would have got a whole new set of reagents, and you would have exhaled through the straw and timed how long it would take to go from pink to colorless. And what do you think you'd expect to have seen with respect to the times? How long would it take to get clear at rest when you're sitting there with your neighbor, as opposed to after you ran up and down the staircase 4 or 5 times? Any idea?
It'd take longer, right?
It shouldn't take longer to clear. It should take shorter time to clear. Why?
Because you're generating more CO2.
Yeah, when you're exercising, running up and down the stairs, there's a higher blood CO2 level produced. And that is manifest in an increased CO2 concentration in the exhalations, which would cause the pH to drop quicker. Does that make sense?
Yeah.
Yeah. And that's, again, what I want you to think about as you read through that sheet and watch that video. Okay. Any questions on lab stuff? Watch the videos, check out the sheets. Quiz next week. Okay, how about questions related to lecture? It could be on the cardiovascular system, which we started this week. I started some voiceover, which I had posted already last Monday. I will continue to put more of them on Blackboard in the next couple days. But I'm assuming you're focusing on the exams, so you're probably reviewing 9 and 19 and 14 more so than 15. But, so open up the floor. Questions, things you want clarified? Would you like to go over related to muscular system, respiratory system, blood?
I just want to point out real quick, I don't know if you know it, the cardiovascular, the voiceover that you did, I stopped hearing anything after like 41 minutes. It just keeps going, but there's no voice.
Wow. I'll check that out. I have no idea why that would be the case. Huh.
Up until that point, everything you hear, but
Wow. And how long was that video? Do you know?
I think it was like 59 minutes.
So there was a couple 15 minutes not being recorded, you're saying?
Yeah. Well,
Huh.
it's being, you're just not hearing the voice.
Yeah, right. Huh.
Would that be, I know some of them videos you put sections ahead for what you're reviewing or going over in the next video. Would that be that section? I haven't finished that video. I had started it.
Oh, yeah. Melissa makes a good point. Sometimes what happens, and it's my fault. Sometimes what happens is I try to do a voiceover for, like, say, from slides 1 to slides 30. Let's say 1 to 30. And then on the system, you've got to specify when you want the recording to stop. And normally I would choose 30. But for whatever reason, my fault, I might have forgotten to change that, and it was defaulting at stopping at slide 45. Okay, so it would have recorded up to where I wanted to, and then I would have shut the system off. But when it goes to the software of the system goes to, save that MP4 file, it's going to save the entire 45 seconds or 45 frames slides worth of material. So that is probably what happened, I'm thinking. Good catch, Melissa. I think that's probably what occurred, because I think I stopped. In fact, I know I stopped after section, what was it? It was the coronary blood flow to the heart. And so it might have very likely, I'm going to go back and check on this, it might have included some slides beyond figure 15.13. Yeah, I bet that's what it did. And so just ignore that point on, that I'm not talking, because as Melissa said, that'll be the beginning of the next voiceover. Does that make sense?
Mhm. Yeah.
Yeah, I think that's what happened. I did that a couple times at the very first chunk of this remote teaching thing, because I wasn't, I was kind of new to the system, and obviously I'm not proficient at it quite yet either, but yeah, that's what happened. We'll cover those slides coming up very shortly. In fact, I'll get working on those voiceovers probably starting tomorrow or Friday or over the weekend. Okay, come on, questions. Somebody must have questions.
I have questions about cardiovascular system, but I'm just wondering if anybody else has more questions about the exam stuff first.
Okay, I appreciate that. Sarah, Michaela, Melissa, Lauren, any questions on muscular, respiratory, blood?
Just looking over my notes real quick to see.
Okay. Because if there's not, then we'll do cardiovascular.
The Billy Verdon and the Billy Rubin, I know in the PowerPoint it had like kind of showed like the structure set up, like I don't know how to explain it.
Yeah.
Like the chemistry form. Are we gonna kind of have to memorize what that is, or is that just,
The chemical formula?
yeah,
No.
okay.
So if we go to okay, let me pull up that. That was the blood chapter, correct?
Yeah,
Okay. All right, so let's do this. Where's blood? Do you guys see do you see a slide that says biliverdin and iron?
yeah,
Okay, so here's the question Melissa was asking about these two pigments, the biliverdin and the bilirubin. I'll kind of flip between these two, or although you can look here on the right hand side too, you can obviously see these. They're really, really, really similar chemically speaking. There are some slight alterations in the hydrogens. And so there's a slight modification basically of the biliverdin as some of it gets converted into bilirubin. But no, you do not need to know or draw out the chemical formula or the chemical structure of these two pigments.
okay. When I went over them, I had written them in my book, but I wasn't really sure.
You do not need to know C33, H34, O6, N4. No. You just need to know what these pigments are, that they are a result of the breakdown of the heme group. So here is, you got to understand what the heme group is. Remember, there's one in each of these 4 polypeptides that makes up the hemoglobin molecule, of which you have millions of these in each red blood cell. So in the center of each of these varying purple shaded structures, these are polypeptides, okay? And in the center is the heme group. And in the center of the heme group is iron. And that's where an oxygen binds. Because you know that red blood cells carry oxygen, right? And some CO2, but typically we think of erythrocytes as transporting oxygen from the lungs to the tissues. So this is taking that spaceship here, looking, flying saucer looking gizmo. That's this. And in the center here is Fe, which is iron. That's where O2 binds. So when you start to recycle red blood cells right after 4 months, you've also got to recycle the hemoglobin inside the red blood cells. And one part of that is taking the heme group, basically breaking one of these bonds, and then these 4 rings just flare out. And you also excise the iron. So you break these little hydrogen bonds between the nitrogens and the iron, break those 2, and that gives you this. That's biliverdin, some of which gets converted into bilirubin. Biliverdin's green, bilirubin's orange. Have you ever, not to get gross, but vomited so violently that you had bile come up?
Yep.
Yeah. Well, the bile was probably green, and basically what you were vomiting up, was a derivative of this. I'm not saying you necessarily vomited up biliverdin, but bile, yeah, I mean bile basically is biliverdin and bilirubin. That's what these 2 compounds become part of bile. So yeah, I guess technically you do. For the green, it's biliverdin. Anyway, so you do not need to know the chemistry.
Okay.
Okay.
How
Any-
long is the exam gonna be?
4 hours. How long?
I mean, like, how many questions are you gonna have on it?
Well, I think we said that exams 1 and 2 were 80, right? And 3 and 4 were 100. So we're upping the points a little bit. I think we mentioned that before, right? So the format's going to be really similar to the other two. The majority of questions will be multiple choice. I can't really ask you matching very well on the Blackboard system, so you won't see any matching. So if you don't see multiple choice, you're going to see either short answer or a couple essays.
Will we still have short essays?
There's no such thing as a short essay.
Well, at the end of our previous tests, you had us take like the short paragraph answer things.
Short
Is that-
answer. Okay. Yeah. Yeah. There's probably going to be some short answer. I would call that a short answer, Joel. You know, by
Okay.
that I mean, you know, you can answer that in less than 5 sentences. That's to me a short answer.
Okay.
Okay. An essay is a much more involved piece of literature, which many students don't quite understand because they see essay as short answer. And no. A lot of people are not writing nearly as much as they ought to for essays. In fact, I don't call a two-sentence answer adequate, but that's what I sometimes get. So I want to make it super clear, not just for me, but for everybody else you take classes from now on. If you see an essay, put some time and effort and thought into coming up with a coherent answer. Okay, people have got to do a better job kind of constructing those. Now I know it's hard now with the online, you know, Blackboard situation because you're having to type it and it's harder, right, to kind of do that. But you know, if you're done with the test and you're, let's say, doing an essay question and you've got 30 minutes left, you know, don't use 5 of those 30 and get done and watch Biggest Loser. I don't mean, spend some time putting that answer together, even if it means writing it out in longhand and then typing it up. I mean, this is your grade. Do a good job on it. So anyway, I know we got off on a tangent a bit there, but there's going to be some short answer. That is, you know, less than 5 sentences or so. And essays are going to involve a little more explanation. The short answer, you know, anywhere from, I'd say 2 to 3, 4 points apiece. What would make a good short answer question? Well, you tell me.
Cardiac system. Well, okay. We're not in the cardiovascular system.
Well, you could use that as an example if you wanted to.
Well, the cardiac cycle, I guess. But
Well, that'd be a good essay question. I mean, that's a pretty big process,
I
right?
guess that would be an essay question.
Yeah.
Yeah.
A short answer might be something as simple as, you know, list 4 of the 5 types of white blood cells. You know, and that would literally be a bulleted answer. It wouldn't even have to be sentences, you know. So
Yeah.
it depends on the question. I consider that a short answer question, even though it's only 4 words. Okay. So, you know, it depends on what the question is. Another good question maybe would be for short answer. Oh, let's see. Muscular system, 9. Describe the functions of the T tubules and sarcoplasmic reticulum. That could be a short answer.
Mhm.
How would you answer that? What is the function of the T tubule and sarcoplasmic reticulum of a skeletal muscle fiber?
transports the calcium.
Which?
Oh, what was that?
Which transports the calcium?
Oh, the T tubules?
T-tubules have nothing to do with calcium.
No. Was that sodium?
No.
Potassium?
The T-tubules convey the muscle impulse from the sarcolemma into the fiber. That's all it does. So there's no ions really flowing through the T-tubules. There's just what we called it a muscle impulse was generated on the surface of the sarcolemma, and then it went inside those little pores. Those pores lead to the T-tubules. Remember that?
Mhm.
And on either side of the T-tubules, you had the cisternae of the sarcoplasmic reticulum. And in response to that muscle impulse, what did the SR do? So the second part of the answer would be, what does the SR do? What's the function of the sarcoplasmic reticulum?
Is that when it transports the ions?
What will happen is the calcium ions will flood out of the sarcoplasmic reticulum. Yes.
Mhm.
Yeah. So the SR, all it does is store calcium. That's really its function. But in response to a muscle impulse, it will release that calcium, and that calcium will bind to the troponin, that three subunit structure on the surface of the actin. And then the tropomyosin in turn moves, freeing up the binding sites. Do you remember all this? This is stuff you need
Yep.
to know for next test.
Yeah.
Yeah. And then when that impulse is done, coming down the T-tubules, and the SR is done flooding the troponin with calcium, and the muscle contraction has taken place, how do we get relaxation to occur? What are the events leading to relaxation? That'd be an essay question.
My mind just went blank.
Doesn't the potassium start coming back in? I'm sorry about the noise, but I think the potassium starts flooding back in and starts and then the calcium floods out, doesn't it?
You've got to go back to the neuromuscular junction,
Okay.
because that's what started this whole thing off, didn't it?
Yeah.
Remember when the nerve impulse came to the neuromuscular junction and the synaptic vesicles released their neurotransmitter ACH across the cleft,
Mhm.
and the ACH bonded to the receptors on the folded sarcolemma, and that's what initiated the flow of sodium into the cell, and that's what generated the muscle impulse that traveled down the sarcolemma into the T-tubules, blah, blah, blah. So you've got to stop muscle impulse generation. How do you do that?
Your body has to generate acetylcholine esterase.
Yes, you have to generate the enzyme. And where is that generated, Sarah? You're right. Where is that produced? Well, where's the acetylcholine at? Because that's what it degrades.
The acetylcholine is inside the folded,
Synaptic cleft.
yeah.
Right. The acetylcholine had made contact with those receptors on the folded sarcolemma. That's absolutely right. So kudos. Good. Yeah. So you've got to remove that acetylcholine from the receptor sites so you no longer generate muscle impulses. Because if you can do that, you won't have the sodium flooding in and you won't have muscle impulses going down the sarcolemma. So the acetylcholinesterase takes that acetylcholine and breaks it apart and removes it from the receptor. And that leads to relaxation, because then are there impulses going through the T-tubules? No. There's no more muscle impulse generation because you've taken the acetylcholine away. If there's no muscle impulses going down the T-tubules, what will the sarcoplasmic reticulum then do? what did it just do in response to the muscle impulse?
Oh, it released calcium.
Release the calcium.
Right, it released the calcium. Now, is it going to release more calcium?
No, it starts
No.
storing it.
It's going to start to actively pump that back. Active transport. It's going to remove them from the troponin, put it back into the SR, waiting for another nerve impulse to release it again, maybe. But we're not talking about that. We're talking about relaxation. So take the calcium, pull it off the troponin, pump it back into the SR. Okay, if you remove the troponin, the calcium from the troponin, what's going to happen to the tropomyosin?
The binding sites are going to be covered up.
Right, those are going to slide over and block the binding sites. Can the myosin come up and grab on and do this ratcheting thing? Nope. What's going to happen to the sarcomere?
It's going to lengthen.
Yeah, exactly. You got it. You've been studying. That's right. Good for you. This is stuff that should be, at this point in time, because we got the exam in just a few days, you should be understanding this. Yep. So you have to kind of make it a story. You have to kind of think about what happens and take it through the whole process, because then it's going to just be easier to understand. And the book does, I think, a nice job, as do some of the videos that I think I posted in chapter 9, of walking you through the different steps, both of contraction and relaxation. Okay. So it's super important that you understand what's going on. So I would spend some time studying that table 9.2. That's a fundamental part of this whole chapter, right? How does a muscle fiber contract? How does it relax? What are the events that lead to those processes? Other questions on muscular, respiratory, or blood?
You said we're also doing joints.
And joints, right. That's right. That's correct. Joints too, right. Right. All right, how about cardiovascular then?
I just had one quick question about that. Was one of the videos said that there's like two types of cells that make up the cardiac muscle? There's like pacemaker cells, and I can't quite remember what the other one was.
Okay. Yeah, this is going to be getting into some topics that we'll be actually talking about. I'll be talking about in the next voiceover when we talk about cardiac control. And I think probably what you're referring to is the fact that there are some cardiac cells that function as a pacemaker to initiate and distribute impulses. Yeah, so it's almost like partial nervous tissue, part muscle tissue. It's a very kind of interesting hybridization kind of tissue. It's in the heart, so it's technically, call it cardiac. But its role isn't so much in contraction like most of the heart muscle is. It would be in the initiation and distribution of impulses that result in contraction. Because every time your heart beats, it beats as a result of what's called the cardiac conduction pathway. And that's what we're going to talk about. I'll be getting into that voiceover coming up in the next few days. It's really interesting, fascinating, I think.
So if the heart kind of makes its own pulses, it's an involuntary muscle. So is it controlled at all by the brain?
It is. Yeah.
Okay.
Even though we talk about it being involuntary, you're absolutely right. And if you think back to that video that I showed you in lab that first day, another segment of that video, I remember clearly showing two heart muscles in a Petri plate, and they were contracting independent of one another, and they had some sound effects in the background. So when the one contracted, it was like a higher beep, and then when the other cell contracted, a slightly lower beep. I don't know if you remember this or not.
I remember.
But what happened when they touched?
They beat in unison.
Right, exactly. So in that sense, they have this very unique ability to sort of contract without any nervous impulses controlling them. But the fact is that in you and I, yes, we have special types of muscle tissue and nervous, we'll call it partial muscle, partial nervous tissue, that helps to set the pace of the heart. But that pacemaker, that specialized piece of tissue in the upper right atrium, is also influenced by nerves too. And that makes perfect sense. If you think about what happens when you're driving down the road at night in the fall, it's a rainy night, it's in October, and suddenly this big buck crosses the road in front of you as you're zooming down 86. What immediately happens to you? You don't hit the deer, thank goodness.
Your heart rate accelerates.
Right. Your heart rate accelerates, your breathing rate accelerates, you maybe start to get sweaty palms. You know, it's called the fight or flight response, right? We've all experienced that. That increased heart rate is due to stimulation by what's called the sympathetic branch of the autonomic nervous system. And the release of adrenaline is the result, and that increases the pacemaker rate of stimulation. So your heart races. Yeah. And then there's other nervous action that can actually sort of depress or slow the heart rate down too. And we'll talk more about that coming up. So that's a long, involved answer to your question, but it is somewhat independent, but it is also regulated by our nervous system as well.
Okay.
Yeah, good question. Good observation, yeah.
I had a question. It's not really important. I was just wondering, why is it like the visceral pericardium? Why is it called the visceral pericardium?
Well, to me, I have to tell you what I think when I consider the term visceral. I think of it being on the surface of. Now, I'm not saying when you look up visceral in Merriam's dictionary, it's going to say that, but visceral can mean all sorts of things. Like, oh, he, in an everyday conversation, we might say, he's very visceral in his reaction to the president's comments. You know what I'm saying? It's a very generic use of the word as opposed to the A&P use of the word visceral, which could mean, the guts, the viscera. But then we talk about visceral pericardium, like you're suggesting. That's kind of a totally different use of the word in that sense. So here's what visceral says in the book. This is right from the glossary. Pertaining to the organs in a body cavity. Now, most of the time, when I think visceral, I think of stomach, small intestine, large intestine, liver. But technically, the lungs, the heart, they're in cavities. They would be considered visceral organs just as much as the stomach would be. So back to the difference between visceral and parietal membranes. A visceral membrane is going to be on the surface of the organ. That could be the lungs, in which case we call it what?
The visceral pleura.
Right. And if it's heart, visceral pericardium. Yeah. If it is on the surface of the small intestine, we would call it the visceral peritoneum. Peritoneum, yeah, is the term we would use for abdominal pelvic organs. And then, of course, what would parietal refer to? And we can look that word up too and just see if it says anything in here. Parietal. Parietal, pertaining to the wall of a cavity. That's what it says in the book. All right, so obviously parietal membranes, be they parietal pleura, parietal pericardium, parietal peritoneum, would be lining the very outer surface of that cavity. So between a visceral and parietal membrane is that theoretical space called a cavity. And in the case of both the lungs and the heart, if you look at those parietal membranes that are lining the outer part of the cavity, they fold back upon themselves to then manufacture, or not manufacture. They fold back upon themselves to become the visceral membrane. So that's kind of interesting.
Can I ask a question about the quiz we took last Monday?
Sure.
I was just unsure about one of the questions. Well, maybe more than one, but there's this question about what does the muscle inside the hip bone do? On the inside surface of the hip bone?
You know, I got to go back and look because I'm not picturing the question. Let me ask you a question real quick, because
Okay.
somebody this morning, Lauren, it was you, I think, or who was it, that indicated that, maybe it was Michaela, that wanted to find the answers to the questions on the quiz, and I had forgotten to put the check marks on to give the correct answer. So when you checked your quiz, the answers weren't listed there, were they?
No.
I don't think so.
Okay.
No, they
so-
were.
No.
All right,
They
so
were.
I just read this before class today, and I went back into Blackboard, and again, I'm sorry about that. So call me on it if you're not seeing it, tell me. I went back in and I did make the check marks that hopefully now when you go back and look at your quizzes, the answer should be listed there. If they were like short answer questions or essays or what have you, I'm going to be providing you with some feedback there as to what I was looking for.
Okay.
And I'm just drawing a blank on that particular question you're asking about. We can certainly go look that up. Let's see, how do I do that? I need to get to Blackboard. Well, I'll tell you what, you go back in and look, and then,
Okay.
and then tell me if it's still confusing. Other questions on the quiz, lecture stuff, lab stuff?
It's not really a question, but I noticed that there was a in the first Arapo videos.
Yeah.
There's a really cool picture of like the inside of the heart. Like I don't know if like it's still a beating heart, but it's, I don't know, there's light in the one side and you can see pretty much everything. It's really cool.
Yeah, I was really impressed with it was Arapahoe Community College video that I believe uses the same textbook that we use. And so since I've been doing these, these voiceovers, I thought having an alternative lesson that you could watch might, it's always good to hear it from two different people, there are two different viewpoints. Sometimes, one person can provide information that the other one, just doesn't think of, or maybe they're experienced in that clinical aspect of it or whatever. So that's why I kind of provide those. So I'm glad to hear that you're looking at those. Some of them are pretty long, and of course you don't need to watch the whole thing. You can just break it up into 15-minute increments or whatever. But yeah, I did look through those. That's why I thought they might be helpful to you. So good. Yeah, I'm glad you're using those.
Do you know which section of Blackboard I would find my quiz in?
You go to the grade center, which is
Okay.
toward the bottom, and then you can click on your quizzes.
Mm,
You should be able to see.
That's what it's called, Grade Center?
Grade center, yeah. What, like there's the content and there's announcements and there's Zoom and all those.
I just see my grades.
Well, maybe that's it then. Maybe called different in mine.
Yeah, on ours it's my grades.
Okay. It's different for the instructor, I guess then.
I have a question real quick on like question 1 of the quiz.
All right, let me pull it up here.
Oh, okay, I spelled it wrong, I guess.
let me go in and look. Here's Blackboard. Okay. No. I should be able to share that. Why can't I see that? Okay. Let's see. All right. So I'm just going to go to Sarah's real quick here.
That's all right.
Okay, you don't mind me looking here?
No.
Okay. So here's Sarah's. Now, no, that's weird because I went through all of these, and I'm not going to not give you a point for that. I mean, that should be 1. This should say 1. I swore I went through all of these because I do look to see whether you missed the spelling, because this is, if you don't put the correct spelling down, it says wrong. The system just looks for the spelling. So you should have got an extra point for that. So go through those, and if it's close and you didn't get the point, ask me about it. That should not have escaped my attention. I swear to God, I went through all of these on Wednesday. I don't know how that happened. So that's going to bump you up then, Sarah.
It happens. I understand.
And then Joe, you happen to be the next in alphabetical order, so this is your
Okay.
quiz. And again, I don't want to show everybody because of privacy issues, unless you don't mind me showing them.
I don't mind.
Okay, so you
Uh-oh.
got number 3 wrong here, but you got those mixed up.
Oh.
And this is showing the outer connective tissue on the
Oh.
surface
Okay.
of the cell fiber, muscle fiber, not the sarcolemma, which is underneath that.
Okay.
And
Thanks. Okay.
and likewise, you should get a point for this. I don't know why I'm gonna have to go through all this and double check them now, because I swore to God I went through all of this and looked for spelling issues. This is giving you 0 points, and you just put a capital G
Oh.
instead of me having the lowercase. Because, as you would write this out in a sentence, there's not a capital G here in the blank, right?
Yeah.
This is not a formal term. It's a noun. Now,
Okay.
the system doesn't know that. So this should be a 1.
Oh, there it is.
Okay, so this is asking about the action of the psoas major.
Mm.
And you put abduction and rotation of the thigh, and the answer was flexion of the thigh. I don't know if you mixed up.
Oh, yeah. Okay, I see that now. That makes sense. It'll be flexion.
Um,
For some reason, I thought it was pointing at the iliacus, like the muscle covering the.
oh, yeah. Did I say psoas major?
Yeah.
Yeah. Hang on a minute here. Let's triple double check that guy. Yeah, iliacus. And if we look on that sheet, what exercise was that?
It was 22 or 23.
Yeah, 23. both the iliacus and psoas major that make up the iliopsoas group have the same action, which is flexion of the thigh. Had you listed gluteus medius, maybe that's what you were thinking.
Maybe.
That would lie on the opposite side. You know what I'm saying? This is showing anterior. Your gluteal butt muscles are behind here. Do you know what I'm saying?
Yeah.
Yeah, so that's what you perhaps were thinking here. The gluteus muscles are abduction and rotation, but that's not what the muscle is here. This is not the gluteus muscle. So. Okay, so that's going to adjust both Sarah and Joseph's grades a little bit, and I'll go back in and I'll check all the other ones just to make sure. Now that we saw a couple that should have been given credit for. I went through this. I don't know why the heck it didn't show that. So that's why you should look at your quiz, make sure that things are copacetic, and if they're not, ask about it.
And mine's the same way with the gastrocnemius.
Okay, I'll go back and look at those then.
Yeah, same here as well.
Oh, that's totally bizarre. I swear to God, I went in and I changed those because I saw a lot of people had that right, and just because they, had a couple letters out of order or capitalized the G instead of not, you should have got credit for that if it's close. So I'll go back and I'll check those when we leave. Okay, any other questions on stuff?
No.
Where did you go to find those lab videos you said were important for next lab quiz?
Go into Blackboard.
Yeah.
Go to content. Go to stuff to copy and bring to lab each week. And then there should be a file down here that says April 20th to 24th lab. That's what we're doing this week. If you're looking at other previous labs, here's the one we did the preceding week, the 13th through the 17th. Do you see that on your screen?
Yeah.
Did you not see these, Joe?
No, I don't know why I can't see them.
You don't see them now?
Well, I'm looking right now on my Blackboard, and when I go to stuff to copy and bring to lab each week, I don't have as long of a list as you do.
No, you don't. That's true. But you should have a blue colored, just like this, a folder that says 913 through 917 lab.
Yeah, that's what I see. Is the 413 to 417 lab.
Yeah.
Is there
Yeah.
supposed to be another one?
Yes, there should be the 420 to 427. Why isn't that listed here?
Yeah, I don't have that in mind.
Okay, well now it should be there. You see it now?
Let me refresh.
Yep, it's here
Yep.
now.
Now it's there.
Okay,
Okay, thank you.
sure. Did you have this one before though, Joe?
Yes, I did.
Okay. Okay, so when you go into that then for this week, you should have these. And I know people have been focusing on the exam and probably haven't looked at these a whole lot. But they are available now. I don't know why they weren't earlier. Okay. Anything else? Okay, well then we'll see you guys, I guess, on Monday. I'll send a reminder out, or I think I already did, with respect to the exam this coming Friday. And I'll send a reminder up as far as those two lab quizzes go for the next couple weeks.
Okay.
Okay, guys, have a good rest of the week. We'll see you next week.
Hey, you too.
Yeah.
Okay,
Bye.
bye.
Bye.